Ladies and gentlemen, it's my great pleasure to welcome you all here today. And it's my even greater pleasure, much as I care about all of you in the room, it is my greater pleasure to welcome Professor Thomas Südhof to HKUST and this Institute for Advanced Studies seminar. Professor Südhof got both his MD and his PhD degree from the University of Göttingen, and then migrated to the University of Texas Southwestern, where he did a postdoc with Drs. Brown and Goldstein, who you may recall were the winners of the Nobel Prize for working out part of the story of cholesterol metabolism, and Dr. Südhof was in the middle of that struggle. He stayed on in Texas and became a member of the faculty there, where he remained till 2008. And then in 2008, he moved to Stanford University, where he became, I need to find out what he is, the Avram Goldstein Professor in the School of Medicine at Stanford University Medical School. And since 1986, he has been a member of the Howard Hughes Medical Institute. I cannot possibly do justice to a lifetime of really superb work. I would say, just from my own perspective, that one of the defining features of what he's been able to contribute is that more than working on a single molecule, he has sort of conceptualized the entire synapse, including its constituent vesicles. And I think, at least as I read his published work, it's that broad and comprehensive view that really has allowed him over the years to make hugely substantive contributions to the field. And as you all probably know, it's not just I that have noticed that he's made these contributions. He has been recognized by many for these contributions. He's a member of the National Academy of Sciences of the United States, member of the Institute of Medicine, American Academy of Arts and Sciences, and has received the Kavli Award in Neuroscience, the Alden Spencer Award, Bristol-Myers Award in Neuroscience, the Pisano Award, Lasker-DeBakey Medical Basic Award, and in 2013 was the co-recipient of the Nobel Prize in Physiology and Medicine, honoring his and others' work on the vesicle trafficking within cells and its importance to both physiology and medicine. His talk today is on the two proteins that have captured his interest of late, Neurexins and their ligands. And he'll talk to us today about their role in synapses and in human disease. And if I may give a spoiler alert, I got a preview of this talk, and you're in for a wonderful ride for the next hour. So without further ado, please join me in welcoming Thomas Südhof to HKUST.
Okay, guys. Thank you for this kind introduction. It's a pleasure to be here. I've been here for the GRC, to say this is a wonderful building. I'd be tempted, actually. I wonder why you're all in here, because when you're outside, you can have this wonderful view over the bay, over the ocean, and it's really terrific. If I had my lab here, I'd probably be spending most of my time at the window, or outside. Well, not now outside. It's too hot outside. But anyway. Some of you may wonder, why am I talking about Neurexins, Neurexin ligands, synapses, and autism instead of vesicular traffic? In fact, I was thinking about talking about vesicular traffic because we continue to be forgotten that I already had given a title that was different. And so that was one of the reasons. Another reason is that when Jim Rothman, Randy Sheckman, and I received, I don't know whether justifiably so or not, but that is a different issue, the Nobel Prize and for sort of working on vesicular traffic, as you can see here, with different types of skills and different types of concentration, as you can clearly see. By the way, that's the only time I've ever been on cover of any magazine, so probably the only time I ever will be on any cover. In some way, that was wonderful, but most of my lab over the last decade, in fact, has been working on a different question. And that question is a question I've been interested for as long as I have been interested in vesicular traffic. But it's nevertheless a question that we have not made that much progress in, to be honest. So what I'm going to tell you today is some progress, but there's much more to do. How many of you are neuroscientists here? But in neuroscience, there is a huge debate continuously about who does the best neuroscience and the most functional neuroscience and the most important neuroscience. And surprisingly, the people who do different things always think that what they do is the most best and the most surprising. So nevertheless, over the recent years, there has been a theme that goes through most neuroscience. And that theme is that one can deconstruct all the problems of neuroscience, all the problems that neuroscience wants to address, which is basically how the brain works, by focusing on circuits. And a circuit is a very, very loose and imprecise term. It's actually maybe not even such a useful term. Nevertheless, we all use it. What you see here is a schematic of a small circuit in the cortex, in fact, which consists of a pyramidal excitatory neuron that connects synaptically to two different types of inhibitory interneurons, both of which then form synapses back onto the original pyramidal neuron. So it's a very typical negative feedback loop, if you want. And it illustrates many features among circuits, among others, the fact that circuits are not circular. What this circuit, for example, does is it modulates the information flow that goes from the inputs to this pyramidal cell to its outputs at axonal outgoing synapses. One can arguably say that the most important component of a circuit are the synapses, which you see here in EM. And these synapses are what makes a circuit, because a circuit is basically formed by synaptic connections between neurons. There's two principal sort of goals, and anything in between, in studying circuits. One goal, as shown here, is to study particular circuits in brain, of which there's many. And this goal tries to answer which neurons are connected to what other neurons in the circuit. What does the circuit do? How does a mouse move, for example, when you make a neuron in a circuit fire at high rate? And this is actually, I would say, the vast majority of all studies in circuits right now. It's basically trying to find out the wiring diagram, trying to find out what happens when you make a neuron fire in a circuit, and how that impacts behavior and so on. Since there's a lot of circuits in brain, billions probably, there's a lot of circuits you can study. And so there will be a lot more papers. And for those of you who are young and starting off their career, it's not a bad thing to go after this, because there's so much to be done. Each one of you can have their own circuit to study and then publish papers about it and be happy. Now, there's a second goal in circuits that is actually what I've been more interested in, which is not to study particular circuits as a goal, but to study what I would call the circuit as such. Sort of the essence of a circuit, very German. And basically the idea here is to look at what are the general molecular principles that mediate the formation, maintenance, and restructuring of all circuits. How are the properties of circuit elements defined, and things like this. It's kind of trying to understand in very principal forms, not individual circuits, but the principal mechanisms that govern circuits. So what defines a circuit? Well, how is, in the way the term is used, how is a circuit determined, if you want? And so when you look at a circuit, which is basically, as I mentioned before, not really a circular thing, but a flow of information through neurons, the input-output relations of a circuit principally depend, of all circuits, of the circuit as such, depend on three properties. One is the intrinsic neuronal properties, that is, how the electrical properties, how does an action potential fire in a neuron? What is the action potential firing threshold? What are the dendritic properties, end and end? Second, synaptic connectivity, obviously, because a circuit, after all, is defined by neurons connected via synapses. I mean, the connectivity, the wiring diagram, the connectome, whatever you want to call it, is a crucial element. And third, something that's often neglected, the properties of the synapses in a circuit. And here, for those of you who are not neuroscientists, I think many, I talk to at least, have the impression that a synapse is basically always the same. That you transmit information at a synapse by releasing neurotransmitters that are then recognized by the postsynaptic neuron. But as it turns out, just like there's hundreds of types of neurons, there are probably hundreds of types of synapses. Synapses differ dramatically in their properties, in the release probability, in short-term plasticity, of which there's many different forms, and long-term plasticity, of which there's also many different forms, and many other things. It is actually not the type of neurotransmitter that is the most variable, since I would say probably 99% of all synapses in brain use either glutamate or GABA or glycine. But it is really the properties of the synaptic transmission of information as a function of the action potential patterns that makes synapses very, very different from each other. So these three properties are what determines the overall properties of a circuit. And the hypothesis that I'm operating under, in order to explain sort of the circuit as such as much as we can, are that the synaptic connectivity and the properties of synapses both are determined by cell adhesion molecules, molecules that connect pre- and postsynaptic sites across the synaptic cleft to each other. These molecules are named cell adhesion molecules because in biochemical assays they can make cells adhere to each other. But most of these molecules are actually signaling molecules that basically function to signal to both sides of a synaptic junction what to do, or any intercellular junction. The second hypothesis is more concrete, namely that cell adhesion molecules called Neurexins and their ligands are central for transsynaptic cell adhesion. And the third one is that the pathogenesis of autism involves changes in transsynaptic signaling, mediated, among others, by Neurexins. So these are sort of the three ideas that underlie what I'm going to tell you about. And I'm going to first tell you about Neurexins and their ligands, tell you what they are and sort of what the principal cornerstones, the principles of their properties. I will then tell you two stories. One about the function of Neurexins on the example of Neurexin 3 and the alternative splicing of Neurexins on the example of what's called splice site number 4. And then a totally different story about a postsynaptic ligand of Neurexins called Neuroligin, and specifically of an autism-associated mutation in Neuroligin 3. Okay, how it all started. So this work actually started at the same time as the work for which I was a co-recipient of the Nobel Prize. Unfortunately, I didn't get it for this one, but anyway. And it started with studies that we performed that were based on this creature here. Does anybody know what this is? Yeah, good. Better stay away from it. Anyway, so this is the black widow spider. And it's one of the most beautiful spiders. For those of you who are female, you will appreciate the mating habits or rituals. I don't know if the males would appreciate it, but anyway. But apart from its somewhat unusual romantic proclivities, this spider produces toxins. A lot of them, most of them, act on invertebrates, but one that acts on vertebrates, and that toxin is called latrotoxin. And this toxin caught my interest very early on because it triggers massive neurotransmitter release from synapses. And it is thought to bind to the presynaptic terminal, and when it binds to presynaptic terminal, it triggers release. And the question that we ask is, how does it do this? What are the receptors for this toxin? And so some years ago, we purified and cloned such receptors. And when we cloned them, we found that they constituted a family of cell adhesion type molecules that we named Neurexins. And this was in 1992, more than 20 years ago. And what we also noted is that these proteins had a highly polymorphic structure due to extensive alternative splicing. And so we speculated, based on their presynaptic localization, their polymorphic structure, and their sequence similarities to proteins associated with neurogenesis, whatever that means, that these might be what we call cell recognition molecules. That is, molecules that are displayed on the surface of neurons. And by binding to different ligands in a manner that's determined by their polymorphic structures, may actually mediate information transfer and specifies or even forms synapses. And after more than 20 years, I think we are slowly getting to the point that we are validating this, and others are validating this this prediction. Neurexins, schematically seen, look like this. They come in two flavors: alpha neurexins that are big, beta neurexins that are small. They're transcribed from the same genes, beta neurexins from an internal promoter, because they are largely identical to alpha neurexins except for a little N-terminal sequence, although they lack most of the extracellular sequences that are present in alpha neurexins. It's a modular domain structure with a lot of LNS domains, 6 in alpha neurexins, 1 in beta neurexins, and 3 EGF-like repeats. There's 3 genes for neurexins in mammalian and mammals. Each has independent promoters for alpha and beta neurexins, so this is a constant feature. With Baroque alternative splicing at 6 canonical sites, which creates thousands of isoforms, binds to intracellular PDZ domain proteins, and many gene mutations have been found to be associated with neurexins, in particular neurexin 1 alpha, in autism and schizophrenia. When we cloned the neurexins, we assumed that they are synaptic because they are receptors for alpha-latrotoxin. But only recently were we able to actually show that they're synaptic, because we have, maybe due to incompetence, been unable to make antibodies to these that really worked well enough to allow localizations. So in somewhat of an act of despair, we made Knockin mice that contain an HA epitope in Neurexin 1 alpha and 1 beta. And what you see here is immunolocalization of these Knockin Neurexins. Nice ringtone, I like it. Yeah. What you can see here is that when you co-localize with a presynaptic active zone marker piccolo, there's a pretty good co-localization. When you co-localize with a postsynaptic marker, there's an adjacent localization. And these experiments, as well as many others, suggest that neurexins truly are synaptic. when we cloned the Neurexins, we found that there was baroque alternative splicing at 6 sites. And using simply combinatorial arguments, we suggested that there are thousands of isoforms. And recent work has demonstrated that these isoforms actually do exist, although there are some arguments whether it's 1,000 or 2,000 isoforms. It's a lot. Suggesting that these molecules are indeed highly, highly polymorphic. Now, as you probably know, in the last 20 years, it's become apparent that virtually every mRNA is alternatively spliced. Alternative splicing is nothing special. When we cloned this, thank God it was special, because that way we could publish our paper well. But nowadays, that is not the case. So one of the stories I'm going to tell you about is testing whether alternative splicing actually means something. And as a first approach, alternative splicing, in order to be in some way have information content, needs to be regulated. And it turns out that most, if not all, Neurexin alternative splicing is actually highly regulated. And I'll give you one example in splice site number 4, which is the 4th splice site. What you see here are quantifications of the total amount of Neurexins, 1, 2, 3 on top, and then the ratio of insert containing to insert lacking mRNAs for each of these Neurexins. And without going into these details, these are mRNAs that were isolated from whole regions, each of which obviously contains many different types of neurons, like the olfactory bulb, cerebral cortex, striatum, and so on. And what you can see is that in these broad brain regions, there's a dramatic difference in the alternative splicing just for this one site here, as measured by quantitative RT-PCR. And for example, if you look in the cortex, there is, relatively speaking, much more SS4 minus, because the ratio is less than 1, than SS4 plus, with a particularly high degree of prevalence for SS4 minus in Neurexin 3, whereas in the adjacent striatum, the opposite is the case. There is a several-fold increase of SS4 plus over SS4 minus for all 3 Neurexins. And in fact, one of the things that's striking here is that all 3 Neurexins are coordinately alternatively spliced in these various regions. So these experiments suggested that alternative splicing is regulated and thus may mean something. Are Neurexins functionally important? After we cloned them, we embarked on a large project to analyze Neurexins by knockouts. And at that time in science, hard to imagine, there was no PCR and knockouts in mice. So we generated constitutive knockouts of alpha Neurexins in mice. And over a long period of time, because these mice bred very poorly, we analyzed their phenotype. And what we found is that single alpha Neurexin knockouts were viable, suggesting that single alpha Neurexins are not essential for survival. But double and triple Neurexin knockout mice largely died at birth. There was, however, no developmental phenotype that we could discern and no significant loss of excitatory synapses in these alpha Neurexin knockouts that still contain beta Neurexins, although there was a massive block of synaptic transmission, suggesting that these mice die because of a loss of synaptic transmission and that Neurexins are important. To illustrate this to you in just 2 slides, when you look at spontaneous miniature synaptic events, you see that both for AMPA receptor-mediated excitatory and for GABA receptor-mediated inhibitory minis, comparing the single knockout to alpha to the triple 1 to 3 alpha. Unfortunately, it wasn't possible to compare wild type in these because it's just impossible to get glutamates in these causes, as you can imagine. You can see that there's a massive impairment in the frequency, but not the amplitude, of spontaneous miniature synaptic events, which is a classical signature of a presynaptic impairment. Similarly, for evoked synaptic responses, here measured as GABAergic inhibitory responses in cultured cortical slices that were stimulated by this 4 action potential protocol as shown here, the initial amplitude, the first response amplitude, was massively impaired by the triple knockout. And the failure rates, which are characteristic of a loss of presynaptic neurotransmitter release, were enhanced. So these mice had the classical of a loss of presynaptic neurotransmitter release in response to action potential. A loss that was caused by, as far as we could tell, a number of molecular impairments such as impaired calcium influx, impaired vesicle priming, and other problems. Now these studies documented, well, more than 10 years ago, that neurexins are truly important. They really are important. But in some ways, these analyses were a dead end because it was impossible to breed enough mice, especially with the mouse costs in the United States, in order to actually analyze these mice in detail. And it was very difficult to analyze controls, littermate controls, because when you have multiple alleles, it just makes it incredibly hard. So we decided at that point to try to change the way how we analyze this, and I'll tell you about one way in today's talk. However, nevertheless, based on the fact that neurexins perform a fundamental and essential synaptic function as demonstrated in these initial knockout experiments, we argued that there must be some kind of postsynaptic ligand that they bind to affect this function, to actually do this stuff. And so over the years, we and others searched for such ligands and were rewarded, if you want, by identifying a large number of such ligands, most of which, if not all, are now known to be physiologically important as ligands of neurexins. This shows only a subset. The first ligands we identified was Neuroligins more than almost 20 years ago. These are constitutive dimeric proteins that have an extracellular esterase homology domain, which is enzymatically inactive. Subsequently, we and others identified LRTMs as high affinity ligands, this GPCRs, and Machina's group in Japan, in a beautiful study, showed that a family of proteins, C1q domain proteins called cerebellins, that bind to postsynaptic GluR delta 2 receptors, bind transsynaptically to presynaptic neurexins. So these studies suggest that there's multiple ligands for neurexins. Some of these ligands interactions are regulated by alternative splicing, especially at splice site number 4, which I already introduced you, and which is located in both alpha and beta neurexins in this LNS domain. And what happens is that the regulation of binding by alternative splicing at this site differs among different ligands, interestingly, suggesting that presynaptic neurexin alternative splicing influences transsynaptic ligand interactions. For example, LRTMs only bind to splice site 4 minus neurexins. Cerebellins only bind to splice site 4 plus neurexins, but not to SS4 minus. And Neuroligins bind depending on the isoform of neuroligin and depending on the isoform of neurexins, either to plus and minus or only to minus, suggesting again that there is some kind of splice code in these interactions. So these findings indicated that splice site 4 in particular may have a role in regulating transsynaptic interactions, and suggested that neurexins have a multitude of ligands that might potentially mediate distinct functions. Of these ligands, Neuroligins may be the most important neurexin ligands. And that is because, to the best of my knowledge, they're the only ones that are evolutionarily conserved. Other neurexin ligands don't appear to be conserved, whereas neurexins themselves are also evolutionarily conserved. There's 4 genes for Neuroligins. Neuroligin 1 protein is only in excitatory synapses. Neuroligin 2 protein only in inhibitory synapses. 3 is in both, and 4 is unknown. And just like for neurexins, there have been many, many mutations associated with autism in particular in Neuroligins. In fact, not only Neurexins and Neuroligins have been mutationally linked to autism and schizophrenia, but also the interacting molecules. These are the Neurexins and Neuroligins, which have been linked to schizophrenia and autism. LRTMs have been linked genetically to schizophrenia. Shanks are likely the most prevalent autism mutated gene, certainly one of the most prevalent. CASC, on the other hand, which is a PDZ domain binding protein that we discovered because it binds to Neurexins, has been linked to autism, mental retardation, and all kinds of things, as have been MINTs, and some others that are not shown here as well. So this is almost like a saturation mutagenesis experiment. There's a pathway here. It's probably not the only pathway in these neuropsychiatric disorders, but at least there's an indication that this pathway, perturbance or impairments of this pathway, predisposes to schizophrenia and autism. So much for my introduction. I hope you now know everything you need to know about Neurexins and their ligands. Let me move on and speak a little faster because time is running out. So let me talk about function of Neurexin and alternative splicing in the case of Neurexin 3. One of the difficulties after the constitutive knockouts of the alpha Neurexins was that we needed to make conditional knockouts. We needed to analyze conditionally mutant mice. But because beta Neurexins are encoded by an internal promoter, a traditional knockout approach was challenging. And so we decided to approach this problem by targeting the first common axon that is shared by both alpha and beta Neurexins. And we decided to focus initially on Neurexin 3 because of its interesting properties. What you see here is a typical domain structure of Neurexin 3 alpha and 3 beta. Exon 18 is the first exon that is shared by both Neurexins, Neurexin 3 alpha and 3 beta. And so we targeted this exon by floxing it and generated a typical knockout mouse. As it turns out, we were lucky in that this mRNA that contains this truncated protein for Neurexin 3 alpha is not stable but degraded by nonsense-mediated decay. And as best as we can tell, there is no protein made. We converted the conditional knockout mice into the constitutive knockout mice and analyzed them. What we found was that these mice did not survive normally. Most of the mice died at birth. With a lot of care, some of them could be made to survive until P21 is shown here. Those that survived, as shown here, have a decreased body weight, and they have a severe behavioral phenotype. They basically are highly abnormal. For example, they have an increased ataxia, as shown here in the open field test, measured by a force plate, and they have a hyperactivity. So clearly, Neurexin 3 alpha and 3 beta in combination are essential. They're important genes. So we wanted to know if there's an effect on synaptic transmission. We analyzed this in cultured neurons from the conditional mice that were then infected with viruses encoding either mutant Cre recombinase, which is the wild-type controlled, or active Cre recombinase, which deletes the Neurexin 3 alpha beta. What we found, somewhat surprisingly to us, in these cultured hippocampal neurons is that the EPSC was decreased by half, but the NMDA receptor-mediated EPSC was not. As you probably know, there's two principal classes of postsynaptic glutamate receptors, AMPA receptors and NMDA receptors. Most synaptic transmission is normally carried by AMPA receptors, but you can selectively measure NMDA receptor-mediated transmission. The fact that NMDA receptor EPSCs were normal means there's normal neurotransmitter release and suggests that this decrease in AMPA receptor-mediated EPSCs was due to a decrease in postsynaptic AMPA receptors. iPSCs were perfectly normal. Consistent with the notion that there's fewer postsynaptic AMPA receptors, the MEPSC amplitudes were also decreased, but the frequencies were normal. We tried to see if we could rescue this phenotype to determine the specificity of Neurexins. We found that Neurexin 3 alpha could rescue it, as shown here. but only if it is a splice site S4 minus. If it is splice site S4 plus, it could not rescue. Similarly, Neurexin 3 beta could also rescue, but only if it's SS4 minus. Moreover, Neurexin 1 beta and 2 beta could also rescue if they are SS4 minus, suggesting that these neurexins are functionally redundant when overexpressed for this loss of AMPA receptor responses, but only if they lack an insert in splice site number 4. This raised the question whether this SS4 minus neurexins actively maintain postsynaptic AMPA receptor levels. And to test this, we simply measured the AMPA receptors by immunocytochemistry. What you see here is the experiment. Basically, you label unpermeabilized, non-permeabilized hippocampal neurons with an extracellular antibody, epitope antibody to GLUA1, and then you permeabilize and you label them for antibodies with PSD95 and the VGLUT1, which is a vesicle marker. What you see here is that the density of synapses, of punctures, is unchanged. The density or the size of the GLUA1 puncture is decreased exactly the same degree as AMPA receptor-mediated responses. There was a trend for PSD95. Presynaptic vesicle clusters are the same. The signal intensity is the same. It's the size that's decreased. And the number of AMPA receptors is also the same, as shown here by quantitative immunoblotting. Why is this? The most obvious answer and the easiest to test answer is increased endocytosis. Without going through the assay, there's a massive increase in endocytosis, explaining why there's fewer AMPA receptors. So viewed together, this is a surprising phenotype. It's surprising because I told you that neurexins are presynaptic, but this is a classical postsynaptic phenotype. It raised the issue of whether Neurexin 3 generally affect postsynaptic function and whether it may be actually a postsynaptic protein. And so the first test towards answering that question that we did was simply analyze cultures from a different brain region, the olfactory bulb, because it's composed of a relatively few number of neurons and because in bulk analysis it has a different splicing pattern in Neurexin 3 splice site 4, as I showed you earlier. And what we found is that when we analyzed these olfactory bulb neurons is that AMPA receptor EPSCs, just like NMDA receptor EPSCs, were normal. So the phenotype wasn't there. But now there was a different phenotype. There was a decrease in inhibitory iPSCs. Postsynaptic responses to inhibitory synaptic activation were massively impaired. But the mechanism for this impairment turns out to be different. It turns out that this phenotype is not caused by a loss of postsynaptic receptors, but by a decrease in presynaptic release probability. Just as one example to show you this, there was an increase in the paired pulse ratio. This increase is classically observed when there's a decrease in release probability, and this is quantified for one particular stimulus interval again here, typically demonstrating that there's a decrease in release probability at this, which causes the decrease in the iPSC. Consistent with this conclusion, in the mini responses, the MIPSC amplitude was normal, but the MIPSC frequency was decreased. So in these neurons, this knockout looks exactly the same as the alpha neurexin knockout. Very different phenotype. So this is puzzling. Knockout of Neurexin 3 in hippocampus causes postsynaptic AMPA receptor depletion. Knockout of Neurexin 3 in olfactory bulb neurons impairs presynaptic neurotransmitter release. It's a paradox, but there's a possible explanation. And that is that neurexins perform multiple functions via interactions with different ligands, as I suggested to you in the introduction, and that the different hippocampal and olfactory phenotypes reflect distinct ligand interactions, which mediate distinct functions then. And that whether or not a neurexin isoform is required for a particular function, as I showed you for the AMPA receptor in the hippocampus or the iPSCs in the olfactory bulb neurons, depends on whether other functional similar neurexin isoforms are co-expressed, and I should add, depends also obviously on the postsynaptic ligands. So when you take this explanation as a guide, How can we test it? And we decided to test this explanation by focusing on alternative splicing of SS number 4 because of this pivotal experiment that I illustrated to you earlier. These rescue experiments which demonstrated that the hippocampal AMPA receptor phenotype could only be rescued with SS4-, but not with SS4+, but could be rescued by all neurexins, by SS4- isoforms. So this raised the question whether SS4- neurexin selectively and actively maintains AMPA receptors. And so we decided to investigate, try to test, try to explore the meaning of total splicing at splice site number 4 based on these results. And we decided to genetically manipulate alternative splicing. When you look at the exon that's alternatively spliced in your exon 3 for splice site number 4, it's exon 20 that's either included or excluded from the mRNA. And this exon has a very unusual splice acceptor sequence, suggesting that this weak splice acceptor sequence contributes to allowing this exon to be alternatively spliced. And so we converted this by homologous recombination in mice to a perfect splice acceptor sequence, as shown here, with the idea that that would make this exon always be included in the mRNA. At the same time, we flanked that exon with LOXP site with the idea that Cre recombinase would then permanently excise the exon so that the mRNA would always be SS4-. And this actually works. What you see here is measurements of mRNA levels. You can see that the total mRNAs are the same, but that in SS4+, there's a total excess of SS4+ mRNA. That is when the Knockin is there without Cre recombinase, and no SS4- mRNA. When you look at the SS4- neurons, there is no SS4-, no SS4+ mRNA, but an excess of SS4- mRNA. So the Knockin causes constitutive inclusion of SS4+, whereas Cre recombinases causes constitutive excision of that exon. And so the control that is Cre recombinase minus in the Knockin is the SS4+. And the active recombinase produces the SS4-. What effect does this have on synaptic transmission? Well, gratifyingly, in a cultured hippocampal neurons, it perfectly reproduces the knockout phenotype. Loss of AMPA receptor responses, same NMDA receptor responses, same iPSCs, demonstrating that it's not the Neurexin 3 that's important. It's Neurexin 3 SS4- that's important. Moreover, the phenotype here again could be rescued, as before, by all SS4- neurexins. So this looks like the Neurexin 3 alpha beta knockout phenotype in hippocampal neurons. What about olfactory bulb neurons, where the 3 alpha beta knockout phenotype is so different? And again, gratifyingly, in the olfactory bulb neurons, the phenotype that's observed in the 3 alpha beta knockout is gone. There is no phenotype. SS4+ and SS4- have no significant difference in either AMPA, NMDA receptor, or EPSCs or iPSCs. So normal iPSCs, and suggests that here either plus or minus works just fine, and that what is happening is that in the knockout, that it's the protein itself, other parts of the proteins that mediates the phenotype, or the loss of other parts of the protein mediates the phenotype. Coming back to the hippocampal cultures, is the hippocampal neuronal phenotype in the SS4+ due to a loss of postsynaptic AMPA receptors? And obviously it is. Just to go briefly through this, synapse density is unchanged, same labeling protocol. Size of the puncta now measured for 2 different postsynaptic AMPA receptors, GLUA1 and GLUA2, is massively decreased in the SS4+, but not in the SS4-, which is the same as the wild type. And this decrease extends over all synapses, as shown here as the cumulative probability plots. Moreover, there's the same increase in endocytosis, which explains the same thing as for the 3 alpha beta knockout. So, thus, these experiments suggest that, should be the opposite way, notice that the SS4 minus variant of Neurexin is essential for stabilizing postsynaptic AMPA receptors, not the SS4 plus. Sorry about that. There's two questions that arise. One is this a presynaptic or a postsynaptic effect? After all, it's a classical postsynaptic phenotype. By all criteria, this should be a postsynaptic protein. And how does it work? To address the first question, we initially used indirect experiments in cultured neurons. What you see here are experiments where we take cultured neurons that were infected either with control lentivirus, become SS4 plus, or with Cre recombinase makes it SS4 minus. And we then use these neurons, cultured neurons, and we transfect isolated neurons with rescue cDNAs. This is control, this is SS4 minus. When we infect the neurons, as I showed you earlier, we can rescue the phenotype completely. When we transfect postsynaptically, there's no rescue, suggesting that the Neurexin SS4 minus does not act postsynaptically. In another avenue, we took the cultured SS4 plus neurons and we transfected them postsynaptically with Cre recombinase, which converts the postsynaptic neuron into SS4 minus. Didn't rescue either. So these experiments suggest it is not a postsynaptic effect, but they represent a fundamentally negative result. I mean, you could argue all kinds of things, justifiably, that these experiments don't really prove the point. So we went in vivo. We decided to use in vivo manipulations to test whether or not this is a pre- and postsynaptic effect, and by the way, also test whether these culture experiments can be replicated in vivo. What you see here is the preparation, injection of lentivirus, of AAVs into the CA1 region of the hippocampus. The AAVs then cause either Cre recombinase mediated recombination or control, and then we record postsynaptically 2 weeks after the injections in acute slices from hippocampal neurons in the subiculum by stimulating presynaptic CA1 inputs. And this allows definitive characterization of presynaptic effects. And the bottom line of this is that when we measured the NMDA-AMPA ratio, or AMPA to NMDA ratio, in the SS4 plus, which is the control, there was a significant decrease. Whereas in the SS4 minus, there was a significant increase, suggesting that indeed in the SS4 plus, postsynaptic AMPA receptors are decreased, and as a result, the ratio goes down, whereas in the SS4 minus, if anything, it's increased. So this is consistent with the loss of postsynaptic AMPA receptors. To measure this directly, we chose yet another electrophysiological procedure, which consists of measuring the synaptic strength using input-output curves. In the interest of time, I won't go into this in detail. What some of you may know is that in the subiculum, there's two types of neurons. There are regular firing cells and burst firing cells. And when you look at this, we can distinguish them electrophysiologically. And what we did is we recorded from these separately, and we measured postsynaptic responses that are entirely AMPA receptor mediated in this paradigm as a function of the stimulus intensity. And just to illustrate this, the stimulus, the synaptic strength that's AMPA receptor mediated goes down with SS4 plus, but is reversed or rescued by Cre recombinase SS4 minus, demonstrating that there is a loss of postsynaptic AMPA receptors upon presynaptic SS4 plus only Neurexin 3. And this is not an effect of release probability because the paired pulse ratio is unchanged. So this basically, I think, demonstrates that indeed in vivo, presynaptic alternation of Neurexin 3, postsynaptic AMPA receptor responses. How does it work? The most logical way how it works is that there's transsynaptic interaction with a ligand. So we simply measured postsynaptic cell surface ligands in cultured neurons that are SS4 plus or SS4 minus or control. Again, synapse density is unchanged. The size of the puncta for LRTM2 one of the ligands I introduced to you earlier is massively decreased in the SS4+, but not in the SS4-. For a neuroligand, that change does not exist to the same extent, although there is a trend that's statistically not significant. For the LRTM2, this cumulative probability plot shows that, again, it has the same distribution over all synapses. So this suggests that it's a postsynaptic ligand that mediates the presynaptic Neurexin effect, not surprisingly, and that LRTM2, which, by the way, directly interacts with AMPA receptors, may be responsible for the effect. So as far as this story goes, one additional question, sort of almost like a bonus that came up in these experiments, was the relationship of alternative splicing to LTP. And that came up because, as you probably know, LTP is thought, classical NMDA receptor-dependent LTP is thought to be mediated by AMPA receptor trafficking. Since Neurexin alternative splicing had an effect on AMPA receptor trafficking, we wonder whether it also has an effect on LTP. Interestingly, we could use the same preparation to test this question, which was particularly useful here because in the subiculum, the pyramidal neurons, the two classes of pyramidal neurons have different types of LTP, which I find fascinating in itself. One type, the regular spiking neurons, have regular NMDA receptor-dependent LTP. The other type, this fast-spiking pyramidal neurons, actually have a presynaptic type of LTP that's completely different. So we used the same paradigm. We tested first the regular type of LTP as shown here. Without going through the details, here's just sample traces, here's a summary graph, and here's just a total LTP magnitude graph. You can see immediately that this SS4+, presynaptic SS4+, basically blocked most LTP, whereas this presynaptic SS4- was permissive. It allowed it. There was no change in the PPR because this is the postsynaptic form of LTP. So this surprisingly demonstrated that presynaptic alternative splicing of Neurexins dictates postsynaptic LTP. What about the other form of LTP, the presynaptic form of LTP? Again, without going into too much details, as you can see here, the amount of LTP was completely the same, independent of what alternative splice form of Neurexin was present. But there was in every case, every genotype, there was a decrease in the paired pulse ratio because, after all, LTP consists in an increase in release probability. This is a presynaptic form of LTP. And that decrease was the same for all genotypes, demonstrating that there's no effect of the presynaptic alternative splicing on presynaptic LTP. Summary. In the hippocampus, presynaptic Neurexin 3 SS4- maintains postsynaptic AMPA receptors by a transsynaptic action. In olfactory bulb, Neurexin 3, as such, both SS4- and SS4+, maintains neurotransmitter release. Totally different phenotypes, essential functions. In the hippocampus, presynaptic Neurexin 3 SS4- is also required for NMDA receptor-dependent postsynaptic LTP. Both maintenance of AMPA receptor and LTP 3 likely acts by binding to postsynaptic LRTMs. These are the conclusions of these studies. And the implications of these conclusions, first of all, for me most importantly, is that Neurexin alternative splicing is physiologically significant. It demonstrates there's a direct, massive effect on synaptic transmission. Second, in the hippocampus, Neurexin 3 alternative splicing mediates synapse specification. This is what I would call as a classical example of synapse specification, where the properties of a synapse are determined, in this case, by alternative splicing. A presynaptic signal can be required for postsynaptic LTP. Most of you, maybe all of you, are too young to remember the LTP wars, but there was a lot of discussion of whether LTP is pre- or postsynaptic. As it turns out, it's probably to some extent both, in the sense that you need to have signals from both. I'm sure that this classical type of LTP is postsynaptically induced and is maintained by AMPA receptor trafficking. but clearly it requires a presynaptic input. In olfactory bulb neurons, Neurexin 3 has a classical release function that is cis-synaptic, that validates the alpha-Neurexin work, and Neurexin function manifests in a context-dependent manner. In other words, it's different between different synapses, presumably because there's a different complement of other Neurexins and of ligands. Obviously, what I've told you is only Neurexin 3, only 2 brain regions, only 1 splice site. There's much that needs to be done. So if you want to work on this, there's a lot that you can potentially discover. Okay. What I've told you then is that synapse properties specifically are determined, among others, by Neurexin 3 alternative splicing. And this was my second part, and it's 4 o'clock. Let me go then to the third part. And the talk, and by the way, this paper apparently was just published, in case, just today or yesterday, I believe. But the starting point for this part of this talk is that multiple neuroligin mutations were linked to autism. And specifically, of the 4 neuroligin genes, there's been several rare mutations in neuroligin 1. There have, to the best of my knowledge, 3 mutations in neuroligin 3. 1 CNV that causes a deletion, 2 point mutations. And there have been many, many mutations in neuroligin 4. And in these mutations, as well as all of these literally hundreds of papers describing mutations associated with neuropsychiatric disorders, raise the question, are these mutations in any way involved in the disease, or is the mutations, the presence of these mutations, purely fortuitous? And you know, this is an important question, because when you read all these Nature papers, you don't actually notice that each individual mutation is never statistically significant. So these guys, when they write these papers, they bunch these mutations into groups, and then they say, well, they're all the same groups, so we can statistically treat them as the same thing. And then they become statistically significant. But these groups are completely arbitrary, because we don't actually know the function of these genes. And so we can't put them into groups, even if we wanted to. So it's all, there's a high degree of fakeness among these papers. And a fundamental issue here is whether any of these mutations in neuroligins or anywhere is actually functionally important. And so what I'm going to tell you about is studies from my lab that date back actually over the last decade, trying to address exactly that question. And so this is the question. Do autism-associated mutations cause a physiological change that might account for the disorder? And if so, by what mechanism? In pursuing this question, we focused on neuroligin 3. It would have been smarter to focus on neuroligin 4 because there are so many more mutations. Were it not for the fact that neuroligin 4 is not conserved in mice. So neuroligin 4 just looks very different in mice, and it's expressed at 10,000-fold lower levels than neuroligin 3, whereas in human neurons, it's expressed at similar levels. So this is one of the cases where mice are just not as good as humans. And one of the few cases. And so this is why we focus on neuroligin 3, because most of our work is still carried out in mice. Okay, and so the approach was to generate mutants in mice that genocopy these mutations. And we generate neuroligin 3 knockout mutants and alpha-5-1c knock-in mutations. And then we performed over the years some comparative analysis of their phenotypes. And in doing so, we encountered a major initial problem. Sorry, I don't know what happened to the writing here, but anyway. In that, although both mutations are linked to autism in humans, their phenotype in mice at the synapse level and at the behavioral level was completely different. There was no commonality. And so from the very beginning, this raised the question whether we can actually even explain autism based on these mutations, or whether mice are a bad way to test this, or whatever. But it was a truly perplexing and puzzling starting mutation and a starting situation. And in particular, the R451C knock-in mutation caused, on the one hand, the destabilization of the protein because it doesn't fold well, so that most of the protein was degraded. But on the other hand, the 10% that were lost in the mutant mice containing this mutation, this 10% of neuroligin 3 caused many changes in synaptic transmission that we simply never observed in the neuroligin 3 knockout. And this suggested to us that in those synapses that we had looked at and those behaviors that we had studied, these two mutations have different effects and sort of cast approach. We and others looked at water maze and found that the two mutations have completely different phenotypes at the social approach and social memory. They had totally different phenotypes in these things. And particularly the social behaviors were interesting in this regard because, as you all know, people who study autism emphasize social behaviors. However, more recently, in the story that I'm going to tell you about now, we considered the possibility that these mutations may be linked to changes in another symptom domain that is associated with autism, namely stereotypic repetitive behavior. And what we in particular noted in the literature is that multiple independent mouse models of autism that have nothing else in common exhibit the same phenotype in one particular task, which is the accelerating rotor rod. We observed some time ago that Neurexin 1-alpha knockout mice exhibit an increased ability to learn on the accelerating rotor rod. Nakatani observed the same thing for a CNV, 15q11 duplication. The P10 deletion caused the same phenotype. The neuroligin 3 R451C knock-in by studies that were from Jacqueline Corley's lab caused the same mutation. The CNT phenotype and the catnap2 knockout also caused a similar phenotype. So we were wondering whether this might potentially be used as a proxy for repetitive stereotypic behaviors. This is the assay. You put a mouse on an accelerating rotor rod, and you make the mouse, in our protocol first, learn how to stay on an rotor rod that accelerates from 4 RPMs to 40 RPMs. And then after 2 days of trials, we step it up a bit and go from 8 to 80, a protocol that we developed initially a couple of years ago for analysis of Neurexin 1-alpha knockouts. And what you can measure here are two parameters. You can measure the initial coordination, how long does it stay on when you put it on first. And you can measure the learning, how well does it learn to stay on. And what we observed is that in the R451C knock-ins, the initial coordination, as shown here, was the same. But they seemed to learn better. They just became better and better and better, whereas the controls did much worse. And these are obviously littermates. And the way how we analyze this is that we put a slope through the learning curve, and that gives us the learning rate. And where that intersects with the beginning, that gives you the initial coordination. And the plots look like this. This is the initial coordination. It's the same. This is the learning, vastly improved by the R451C. So this is not a loss of function phenotype, which is a somewhat of an advantage. It's a gain of function phenotype. These mice do better on this task. And if you accept this as a potential proxy for repetitive behaviors, it means they learn better how to do the same movement over and over again. What about the neuroligin 3 knockout? Well, obviously it has the same phenotype, otherwise I wouldn't be talking about it. Same initial coordination, higher learning rate. So this is the first behavior that we have observed where the two different types of mutations have exactly the same phenotype. Is this really a stereotype behavior? I don't know if it is, but it has at least some features. We analyzed the movement of the mice using videotaping. You can analyze step timing, step length, and step location. When you measure how well the mice learn to do always the same thing, decrease the standard deviation of the location or the length of, or the timing. In each case, the knockouts do great. They actually learn how to be stereotypic, whereas the wild type have some variability. What about other stereotype behaviors? They're classically labeled stereotype behaviors such as grooming, in which these mice have no phenotype. Now you can argue, is that really a stereotype behavior? What is a stereotype behavior? Argue with somebody else. I don't know. But we are doing these assays to look for things which actually are in some way different. What we found in the open field test, which is another way where people classically analyze stereotype behaviors, is that they had an increased activity. They were hyperactive, both types of mice. And when we looked at the pattern of this hyperactivity, we found that they had an increase in low mobility bouts, variations, they had an increase in hind limb jumps, and they had an increase in rotational bias. And to be honest, we took this from the literature because that's what the literature says, that this is an indication of stereotype behaviors. I don't actually quite understand why, but anyway. So what these data suggested then is that these two different mutations, after all, do have a common phenotypic signature in behavior that is a gain of function, increased learning ability on the rotor rod associated with hyperactivity. And we were interested to understand the mechanism of this behavioral change. We have a gene and a molecule, which is neuroligin 3, and we have a behavior, which is a gain of function, a better learning of a motor task. And what we wanted to understand is how they're related to each other, how this is connected to that. And so in order to understand this, we need to manipulate different areas of the brain specifically. And so we created a conditional knockout mice in which we floxed one of the 5' axons, as shown here, actually 2 of them, which makes the deletion of neuroligin 3 conditional. And we then ask whether the motor learning phenotype is due to a change in the cerebellum or the striatum. And the reason for asking this question in that way is that in classical literature, motor learning is thought to be associated with 2 structures that form loops with the cortex, which is the striatum and the cerebellum. And so we first analyzed the cerebellum. We crossed the conditional knockout mice with L7 Cre mice that delete selectively the neuroligin 3 in the Purkinje cells of the hippocampus, which are the only output neurons in the cerebellum, which are the only output neurons of the cerebellum. And we found no phenotype on the rotor rod. So clearly the cerebellar neuroligin 3 was not involved in this, although we did find an increased activity, hyperactivity, consistent with the notion that the cerebellum, just like the striatum, is an obligatory sort of loop for the cortex. What about the striatum? Now, as many of you probably know, the striatum has 2 principal cell types, in addition to minor components. And these are inhibitory projection neurons, MSNs, that express either D1 dopamine receptors or D2 dopamine receptors and are red or green. They have different projection properties and different functions. I won't go into this. Suffice it to say, there's 2 classes of MSNs that account for more than 90% of all neurons in the striatum. And so we crossed the neuroligin 3 knockout mice, conditional knockout mice, with Cre mice that express Cre recombinase only in D1 striatal neurons. And this completely replicated the phenotype. Increased motor learning, initial same coordination, increased hyperactivity. We then crossed them to Cre mice in which Cre is only expressed in D2 MSNs. This is the A2A Cre. It's not a D2 Cre because the A2A Cre is more specific. And when we did that, we found no phenotype. So in some ways, we found the cell type that is responsible. Why, however, is it D1 but not D2 MSNs? Why not both? So as you probably recall, any knockout phenotype that you observe never tells you the complete function of a protein. It only tells you the function of the protein where it is limiting, where there's nothing else to compensate in some way or another, obviously, right? So we ask whether neuroligins are equally expressed in different types of D1 and in different types of MSNs. And in order to do that, we measured mRNA levels in isolated neurons using fluidigm technology, quantitative RT-PCR. What this shows is that this works. The D1 MSNs express D1 markers. The D2 MSNs express D2 markers. When we did this to measure neuroligin levels, we found that neuroligin 3 was specifically expressed in the nucleus accumbens, the ventral striatum, but not the dorsal striatum, at a higher level in D1 than D2 neurons. Let me just explain this a little bit. This plot shows the ratio of D1 to D2 expression for the different neuroligins. It shows the ventral and the dorsal striatum separately. This ratio is the same for neuroligin 1 and 2 in both types of the striatum. It's almost the same in the dorsal striatum for neuroligin 3. But it's vastly more, since this is a logarithmic plot, in D1 than D2 for neuroligin 3 in the ventral striatum, the nucleus accumbens. So this is great because it explains why in D1 there is this selective phenotype. But at the same time, it's actually not that great because for those of you who know about the role of the striatum in motor learning, it's the dorsal striatum. It's not the ventral striatum. It's the wrong part of the striatum. It should be here. So we were wondering, maybe neuroligin 3 performs a role in the ventral striatum, in the nucleus accumbens for motor learning, that had previously not really been appreciated. And so we used a totally different strategy. We basically knocked out neuroligin 3 in the total ventral or total dorsal striatum using AAV injections, stereotactic AAV injections, just as I showed you earlier. And what you see here is the expression of the AAVs in the ventral striatum and the nucleus accumbens, and this replicated the phenotype. Increased motor learning, hyperactivity, no change in coordination. Then we did the same thing for the dorsal striatum, no phenotype. So clearly, neuroligin 3 acts in the ventral striatum to mediate this effect. Now, this way, multiple controls. I'm not going to go into them, especially since it's getting late. Let me just tell you that you can rescue the phenotype by simply reintroducing into the ventral striatum neuroligin 3, which totally rescues the phenotype. Almost done. Story thus far. So neuroligin 3 functions in a specific cell type and a specific subregion. D1 MSNs, ventral striatum. The function and dysfunction is in the mature circuit, not developmental. This is actually an important point because all these disorders are always referred to as neurodevelopmental. At the same time, genes like neuroligins do not only do development, they're expressed throughout life. The fact that you can both induce and rescue the phenotype in adults means the phenotype is not due to a change in development. It means it's due to a continuing function of the protein throughout the life. Surprising role of nucleus accumbens and not the dorsal striatum of cerebellum repetitive motor task. Are these changes induced by a specific synaptic impairment? Have to be, right? After all, we work on synapses. And they probably are. So we first looked at excitatory synaptic function. And we thought it would be excitatory synaptic functions because there's many papers that suggest that all autism is linked to mGlu-R function. And so what you see here is that there's no change in minis, either D1 or D2, but there's also no change in mGlu-R-mediated synaptic plasticity, very different from what had been predicted. So this was completely negative. We then went on and looked at inhibitory synaptic function. And there we observed a large decrease in the frequency of inhibitory minis. but not in the amplitude, and this change was specific for D1 and not present in D2. So there was a specific change that suggested that D1 neurons, and by the way, we only saw this change in the ventral striatum, but not in the dorsal striatum, again correlating precisely with the behavioral analyses. And so this suggests that there's a change in the inhibitory-excitatory balance that drives the behavioral phenotype, and that this change is due to decreased inhibition, specifically in D1 type neurons in the ventral striatum. And obviously, you can measure this directly. You can record postsynaptically both excitatory and inhibitory evoked responses by changing the postsynaptic voltage clamp. What you see here, if you do this, is, let's just focus on this here, that the knockout causes an almost halving of the inhibition-excitation ratio, a massive change. This is in D1. D2 doesn't have it and shouldn't have it based on everything I told you. So it's highly specific for that. So the model here is then that the behavioral change, we can discuss afterwards, if there's time, what the evolutionary implications are for this. But anyway, this ability is due to an increased excitation or a decreased inhibition-excitation ratio in D1 neurons of the nucleus accumbens, very selectively. This does not mean that other brain regions do not have an impact on these behaviors, but it does mean that in this particular model, that particular synaptic change can account for this particular behavioral abnormality. And so this would be the wild type, where you have a balance like this in the trial, and in the mutant, you have a shift in this balance. So summary, I'm almost done. Both global and Neurexin 3 knockout and 5-1c mutations cause enhanced by decreased synaptic inhibition on D1 MSNs in the nucleus accumbens. This, to the best of my knowledge, is the first localization of a specific behavioral change to a particular synaptic input. And the nucleus accumbens controls repetitive behaviors, whereas dorsal striatum is essential for motor coordination. In other studies, we found, obviously, that everybody else is perfectly right, that the dorsal striatum is absolutely essential for motor coordination. There's many new questions, which I won't discuss. For example, what is the overlap between enhanced repetitive behaviors and reward-related behaviors? The nucleus accumbens is classical, the structure that mediates reward-related behaviors. And how relevant is the synaptic and behavioral mechanism described here for autism? I don't know. So, to summarize, our results illuminate an autism-related behavioral phenotype. They imply that Neuroligin 3 is selectively essential for synaptic strength at a subset of inhibitory synapses in the nucleus accumbens. Our results identify a specific synapse at which a loss of the synapse-specifying function of Neuroligin 3 produces a particular phenotype. But they don't identify a circuit. I think there's many neurons that contribute to this behavior. I don't know how many neurons. I think that we use the term circuit way too loosely. And finally, our results generally reinforce the notion that Neurexins and the Ligands control synapse properties via specific interactions. And so I hope that from this lecture, you will take home, or wherever you go from here, maybe to dinner, this notion that synapses, in general, are built by interactions between pre- and postsynaptic neurons that involve signaling, which in turn is mediated by transsynaptic excitation. And that the central component of that transsynaptic excitation are Neurexins and their ligands that act in many different ways to control different components of this transsynaptic excitation. There were many people who contributed. Most recently, Jason Aoto did all the work on Neurexin 3 I discussed with you together with Katsuhiko Tabuchi, who now has his own lab. He's actually a professor in Japan. Patrick Rothwell and Mark Fucillo did all the work on the Neuroligin 3 that I discussed with you. And Stefan Maxeiner made the mice. Theo did the initial immunocytochemistry that I showed you, and Schaber worked on some of this data on Neuroligin 3 mutations that I haven't really discussed. Thank you for your attention.
Questions? So, please. I'll leave it to you to choose among them.
Go ahead, please.
Okay, so you mentioned that in the SS4 plus mutant mice, the LTP is obviously reduced. Did you look at the behavior of this mice?
No. So the SS4 plus, we haven't studied the behavior. I think, the problem is we do a lot of behaviors in this situation because usually reviewers ask for it. But I think it's pretty meaningless because, it's everywhere. The mutation, you can't, so because it's SS4 plus or SS4 minus, it's everywhere SS4 plus. So in other words, the behavior, the changes in behavior of SS4 plus alone are meaningless. What would be meaningful is if you changed it in one particular brain area, you got a change in behavior. And that's something we would like to do. If you can actually localize it to a specific synapse, yeah. But I agree with you. It's actually, the whole LTP field is a mess. Nobody cares about LTP anymore. People you're so excited, 10, 20 years ago, but nowadays nobody cares. But the fact is, we know nothing about LTP. We don't know how it's mediated, we don't know the mechanisms, we don't know behavioral implications. We have no idea, because everything that was done previously was done with manipulations of chemokine kinase 2 or calcineurin or NMDA receptors that function in virtually everything. So if you manipulate them, you never know whether what you see has anything to do with LTP. It could be something else that's happening. There's just no idea. So I hope that at some point the interest in LTP will come back, because we actually don't know much about LTP, and certainly nothing about its behavioral implications.
Tom, wonderful talk. Too much to absorb in one lecture. So I don't know whether I understood it correctly. So you alluded to that Neurexin neuroligin connections are the chief sort of orchestrators for synaptic adhesions and underlying the functions. And the other types of like LRTM, lateral filling, and so on, are they quantitatively less in abundance, or?
I have no idea, to be honest.
Okay, so the related question, what do you think about, for example, are the distributions or binding partners with Neurexin with postsynaptic ligands, are they shifting in different activity dependent?
It's an excellent question, and I would like to think that that's true. But I don't also know that. So there's many issues here. We don't actually understand Neurexins at this point. What I showed you are indications, sort of like little flashlights, that, this is important for that. But we have no overall view of what they do and what they don't do. They're clearly not the only transsynaptic cell adhesion molecules that are very important. There's others as well. So there's a whole zoo, if you want. And we don't know the relations and the relative importance either. I would be happy and gratified if there was an activity-dependent change. And I hope there is. But I don't think there's any data at this point that support this notion. So there's papers that suggest that when you excite neurons, that alternative splicing of Neurexins change. And they are quite promising, but not totally persuasive.
But if I may follow one more, you showed that thousands, precise number is arguable, but it doesn't really matter, right? Thousands of different splicing forms of Neurexins. Mm-hmm. So are you predicting, so I thought intuitively to encode the very, very many different types of synapse activities, are you predicting there will be, hundreds, thousands of different partners for?
No, but that is actually, again, a good question. My model would be that, so for Neurexins, first of all, the architecture of Neurexins is actually very intriguing, because the beta Neurexins are simple. The alpha neurexins are complicated. One of the things we need to understand is the relative importance of what they do, which we don't understand. The ligands that we discussed up to now are all alpha plus beta, either. But there's also ligands of alpha neurexins that beta neurexins don't bind to. If we just focus only on the ligands that bind to both alpha and beta neurexins, and let's say, just for the sake of argument, that you have 4 different ligands that are all exhibit distinct affinities for different splice variants and different isoforms. By combinatorial mixing and matching, you only need 4 ligands to get thousands of types of synapses. So you don't actually need that many ligands. Yeah. And I think so my hypothesis is that these ligands compete with each other. And a ligand will win over another ligand depending on its concentration, and B, the relative affinities that is dictated by isoforms alternative splicing. And it would be extremely attractive if that could be changed as a function of activity. Yeah. And I would love to see that. Absolutely. It's a great idea. Yeah. So, but I'm not sure we have the evidence to say that that's the case.
Okay, maybe one more question.
Okay. My question actually almost follow up to your last question. Is that I think it's a very attractive idea to have a limited amount of number of interaction between the presynaptic, the neurexin, different splicing, and the postsynaptic, the ligand, and that would, further affecting the receptors, such as the AMPA receptor and the endothelin receptor. Now, the interesting thing here, now you give a lot of good evidence in the part 2 of your talk about this synaptic action. But how much do we know about the actual molecular mechanism, how they interact? Because the ligand is, how the neurexin will interact with the ligand, and the ligand interact with the receptor. Whether they, modify the lifetime in the membrane, they recycle it faster.
Great question. So we know a lot about actually not a lot. There's a few crystal structures of the complexes of neurexins with neuroligins. That's all. We know nothing about the postsynaptic cell biology. And, I've been traditionally only associated with the presynaptic side. The Nobel Prize was for presynaptic neurotransmitter release. But so it sounds almost like I'm hypercritical if I say that the postsynaptic side is disappointingly behind. But the fact is that we know tons of proteins postsynaptically that are important, with literally, I don't know how many papers on them, like Schenck and PSD and whatever. We don't know for a single one of these proteins what it really does. To this date, we don't actually know whether AMPA receptors are inserted into the PSD. So the postsynaptic field, they always say that that's not possible because they think that there's this density and the AMPA receptor vesicles can't go through. But the fact is that presynaptically there's a density too, and the vesicles go through just fine. I have no idea. So this is actually something that I'm trying to persuade people to work on in my lab and in others. And I think it's something we need to know, but I don't know what the mechanisms are. I don't know how LRTM interacts with AMPA receptors, for example. Okay, guys.
All right, and with that, please join me in thanking Dr. Südhof.